We, as you all know, which is part of why you are here, we are in a new age of science and technology. And one of the biggest transformations of this age is that the sequencing of the human genome has turned biology in many ways to an information technology. When computers were first developed, people didn't think about hacking into computer systems. People hadn't internalized this idea, things like Moore's Law. But over time, we've come to expect that our technology expands, the power of our technology grows exponentially, and that IT systems are inherently hackable. Now that we are turning biology into code, which is what sequencing of the genome has done, increasingly biology is becoming an information technology, and therefore it has the same qualities as any information technology, which is exponential growth, at least in some ways, and we've seen that with the cost of sequencing, the power and cost of sequencing the genome decreasing at 2 times the rate of Moore's Law, and hackability. And that's very, very important. And so we are in this age where all kinds of science are becoming informationalized, and they're on this exponential J-curve. And what it means is that our sense of timing, of how long innovation takes for us as a species, is going to change. Because right now our brains are designed for linear thinking. If you're living on the savannah and you have exponential thinking, if you're thinking 10 generations ahead and you're not thinking about the tiger that's coming to get you, you're in trouble. And that's why our brains are designed for linear thinking. But right now, this pace of innovation is going to increasingly get faster and faster. So when we think of maybe 1 unit of change is 2006 to 2016, it could be that 2016 to 2026 maybe 3 times that, 3x. And 2026 to 2036 could be 10x and then 50x. So the speed of innovation is going to increasingly expand. And therefore, our thinking, how we think about the world and how we understand the world, and the things that are fixed and the things that are unfixed are going to change. They're going to be brought into these challenges to the way that we think. And bringing that back to aging, we see these examples of aging in nature where there are a lot of, and we're going to talk about this, a lot of disparities. And we don't fully understand why it is that a mouse in captivity lives maybe 4 years and a mouse in the wild lives 1. And we have some good ideas why an average mouse on the street here, that a bat can live 30 or almost 40 years, why there are sharks that can live 500 years when there are other sharks that are genetically similar that live much less, and why there are jellyfish, and this is in my novel, I talk about this as well, but jellyfish that seem to be immortal, that go back and forth between mature age and a polyp stage. And so we're at this phase in our evolution as a species where we have a question. And the question is our mortality fixed? Is our biology fixed? Or are we hackable? And is mortality in one way or another, or in pieces along the way, overcomable? And hackable. And that's what we're here to discuss tonight. And we really have the super all-star team of people to talk about, as we always do for events at 92nd Street Y. And to my immediate left is David Sinclair, who's a professor of genetics at Harvard Medical School and co-director of the Paul Glenn Laboratories for Biological Mechanisms of Aging at Harvard Medical School, the founding director of the Glenn Center for Aging Research. He's published 130 papers, has many patents, and is co-founder and co-chief editor of the scientific journal Aging, and appeared in Time Magazine's list of the 100 most influential people in the world. And that's very influential.
We aging researchers have been struggling to define aging for the last 30 years, and we're getting better at it. The good news is, though, that we don't need to know actually what causes aging to be able to do something about it. As we'll talk about, I hope, we found many of the genetic pathways that control the pace of aging, from yeast cells through to humans, and the race is now on to control those. Although if we're in the mood for telling jokes, what I say is aging is a lot like pornography. Mm-hmm. You know, I don't know what it is, but I know it when I see it. And that's really true. We know it when we see it, but just being able to describe it doesn't do science justice. We have to understand the molecular pathways, and that's really where we've come over the last 20 years.
What do you see, based on what we know now, which you've just said is imperfect, as the mechanism of aging? What is happening?
Well, there's a lot that goes wrong, just as a machine falls apart as it gets older. But the good news is, if you use an analogy of a car, we used to think that our bodies were just like cars that would wear out and break down. But so the idea of having a single pill to slow down or even reverse aging was ludicrous with that model or that analogy, because it'd be like having a magic wrench that you throw at the car and suddenly all the paint's fixed and the rust is fixed and the engine's fixed. That's never going to happen. But our bodies are much more complicated than cars. We have self-healing repair systems that we now know how to control with small molecules, and that's really the important thing. If I was to put a bet on what actually is the upstream cause of aging, though there's a lot that happens, including what Anna Maria works on, I think it's actually that our cells are what I call losing their epigenetic resilience. And that's just a complicated way of saying it's not the DNA information, the code that's being lost. That's the current idea. I think it's the cell's inability to read the right genes at the right time, so that our cells eventually forget their identity. And so liver cells start behaving more like other types of cells, and we get dysfunction that way. The good news is, if we still have that information, like a DVD or a compact disc that is just scratched, we should be able to reverse aging just by polishing it and allowing the cells to read the information that's in our genome. And one way to think of it, Jamie, is that I talked about those cars, that our bodies are like cars that get older. mTOR is one of those repair body shop mechanics, and what we're learning to do is to get them out of bed earlier in the morning and work harder to repair our body and keep it pristine.
So we're going to talk a little bit later about interventions, and I think everybody has, and we've already started to talk about that. But without even intervening, when we just look at the animal kingdom, there are just differences in aging of things that you'd think, like a mouse and a bat, you'd think, well, they look pretty similar. You'd imagine that they would have a relatively similar lifespan, or different kinds of sharks that look roughly the same, or different whales, but the actual life expectancy is drastically different. David, from looking at variations across the animal kingdom, what can we learn about the different processes of aging?
Well, so for a start, we've been very fortunate to have these lab organisms that we can, as geneticists, find mutants that live longer and figure out what are the genes that control lifespan. That's how we found genes like mTOR and others that control lifespan. But what's great about the whole panoply of life on the planet is that some organisms live a day or less, and some organisms live 300, 400, even if you talk about plants, more than 1,000 years. So what does that tell us? That tells us that, first of all, each species has a certain range of lifespan that must be encoded in the genome. And if you're lucky and you live a healthy life, you can maximize that. But there seems to be a genetically encoded limit. Say for humans, it's about 120, 122 is the oldest of all. And you can exercise as much as you want and eat a perfect diet, and that's about as good as you can get. But what that doesn't mean is that that's our actual limit. It means that, sure, we can live according to our genes, but if we have enough technology, we can fly. It's not in our genome to fly. We can cure diseases with antibiotics, and we can intervene in aging and get us, maybe not living forever, but giving most of us a chance to live into 90, up to 90 and 100 in a healthy way, playing tennis and raising the great-grandkids. And what it actually tells us is, the other animals that live longer than us, is that living a long time is not against the laws of physics. It's doable. And we just need to figure out what the secrets are.
So everyone's talking about calorie restriction. David, why does it work? What's the mechanism of calorie restriction? It's connected to what Anna Maria was saying.
Yeah, sure. The old idea was that calorie restriction just reduces your body's metabolism and you just live slower. But that's not actually true. It's been disproved. And what we think now is that it puts the body in a state of what's known as hormesis. Have any of you heard of hormesis? Raise your hands if you've heard of this word. So
if you raise your hand, he cannot see you.
Yeah, exactly. So you may as well raise your hand.
So anyway, so hormesis is a concept that was actually first discovered by when people were spraying herbicides on plants. And if you dilute it down very far, the plants actually grow better. And another way to think of hormesis, it's a mild stress that doesn't kill you makes you stronger. And by stress, I don't mean the most recent election. I mean biological stress, putting your body in a state of perceived adversity. So running will tell your body that there's adversity. Being hungry puts your body in a state of perceived adversity or hormesis. And the body mounts a defense response. It starts the cleaning process. It turns this mTOR pathway on and other genes that we work on. And then they take care of the fixing process. So essentially what you're doing is, with this analogy of this car repair shop, what you're doing is having a bullhorn and saying to the mechanics, get to work, there's an emergency, we need to build this car. And that's what being hungry and exercising does. And it's not that your blood flows better or being hungry burns the fat and that's what's good. It's actually the body gets into a defensive state that can heal.
So we talked in the green room about swimming in freezing cold water, which apparently has some positive effects. We won't go into that, but if you have freezing cold water and you'd like to go swimming in the morning, you should do it. But can you each talk about the most promising scientific approaches to? I mean, you've all mentioned a little bit. What do you, just going from David across, what do you think are the most promising? I know you've worked on resveratrol and some other things. What do you think? What are the most promising scientific approaches to life extension?
All right, I would put my money on these control mechanisms, these body shop repair managers, and there are really three main contenders. There's a whole network of them, of hundreds of them, but there's three main pathways that we like to talk about. One is, we've talked about mTOR, Nir brought that up. One is AMP kinase, which Nir also works on and is hoping to do a clinical trial to show that can extend lifespan, or at least reverse some aspects of aging. And the third is a group of enzymes that I work on called sirtuins, and there's 7 of those in the body. And I like sirtuins, but they're all equally, I think, valid, and if you tweak one, the others change. So actually, we're all really holding the elephant just from different ends. If you're wondering what the latest thing out of my lab is, very briefly, we know that these sirtuin enzymes protect the cell, and that they need a little molecule that's got the name NAD. And we lose NAD as we get older. By the time you're 50, and I'm almost 50, we have about half the levels of NAD that we once had, and without NAD, you're dead within 30 seconds. So we're developing molecules that are going into human trials next year that boost NAD levels back up to youthful levels. And we see, at least in mice, remarkable effects that mimic diet and exercise.
In our earlier sessions in this Homo sapiens 2.0 series, we've talked about the genomics revolution. And the last session we did was on the future of fertility and in vitro fertilization and embryo screening. And so, theoretically, at least, as we learn more about the genetics of aging, it seems that we're going to be able to predict, based on the sequencing of pre-implanted embryos, which embryos have a greater likelihood to live longer and not. Would you agree? And do you think that if we start selecting embryos to eliminate disease, we'll be able to select based on estimated longevity? David?
All right, so the interesting thing about, there's a lot of questions there, but but on fertility, what's interesting is that what we've recently discovered in science is that females are not born with a set number of eggs. If you see that in high school textbooks, rip that page out, it's wrong. In many mammals, they produce eggs throughout life and through stem cells. And so we and others are growing stem cells from women's ovaries and mouse ovaries. And we can isolate these, we grow them in the lab, we can genetically modify them. And we have the technology, if we really wanted to, though we're not going to either modify or screen these cells for different traits or remove diseases like Huntington's and breast cancer. That's all in the future. One of the other things that's interesting, though, is that the same molecules that we're talking about tonight that reverse aging in adults also seem to work effectively on the reproductive system. And we have some early results that we haven't yet published, but between friends I'll tell you now. We can take an old mouse, the equivalent of a 70, 80-year-old human female, and make that mouse fertile again and reverse what we call mousopause.
Yeah.
Now, think about what that'll be like in a society where you can put off having kids till after you've established your career and your marriage, and but that's the world where we're headed. And getting into questions about selecting embryos for this and that, I'm not really into that part of it, but I do think being able to prevent genetic diseases is something that's going to be much more routine in the future.
Yeah, but if we were doing that, and we've had other sessions on this, and I write a lot about this, conceptually, we would be able to choose based on a genetic fingerprint, if you will, of longevity?
Of course we could.
Yeah.
The technology is almost there already.
Yeah. Anything to add on a dream research agenda?
Well, my dream is that we can accelerate this research.
Yeah.
I know that it's a matter of when, not if now. We have the technology, just a matter of translating this, and people like Near are going as fast as we can. You'd be surprised, I think most people would be surprised that a fraction of 1% of the research budget of the federal government goes towards the basic fundamental biology of aging. So we as researchers are suffering, we're laying off people, and given that trillions of dollars could be saved in the economy, it's quite depressing, and probably will just get worse over the next 4 years at least. But research agenda, I agree with Anna Maria that being able to measure yourself and optimize yourself even before you get sick is important. For too long, we doctors, these two are proper doctors, I'm just a Ph.D, but we scientists have worried more about sick people than preventing disease. But we have the technology now, even from a simple blood test, to be able to say, where would your optimal be? Not just are you out of the range, but let's see if we can give you this or that and tweak you so that throughout your whole lifespan you're optimal. And we can actually estimate your biological age. And through using this biotracking method, and it's through right now a blood test, I was able to ostensibly reduce my biological age from 58 to 31 within a year.